Okay. Hello everyone. I guess we just have one participant as of now, and soon I hope others will also join. So welcome to session 5 of bioengineering and interface with biology and medicine. So this is one more live problem solving session. We'll be discussing topics on chromosomal basis of inheritance and week 5 contents, and I will be taking up some sample questions, and along with it I'll also discuss some concepts behind. So I'll begin the class for today. And yes, this will be a session of 40 minutes each, three sessions of 40 minutes each. And once 40 minutes is over, you will have a message on the top of your uh, screen. You just need to leave and then again rejoin in a minute, and then we can continue. Okay. So just inform me if my slides are visible and they are changing or not. Are they changing? Okay. As there's no response, assume that it is visible and it's changing. Okay. Hmm. So the first question was match the following. These are the different organelles present in a cell and their respective functions. So if we look into the different organelles that are given in the question, they're nucleus, plasmid, membrane, sex pilus, and cell wall. So this is sort of a straightforward question. So for nucleus, plasmid, and cell wall, the answer is pretty easy. That is, nucleus is the basic genetic material. We know it is made up of DNA, RNA, and it has different proteins which are which participate in central dogma. It is actually the main genetic makeup of the uh, particular cell, and all major events are carried out by it. So nucleus is being the basic genetic material. Plasmid is an extra chromosomal DNA which is also which present along with the nuclear material, but it doesn't always integrate into it. It is separately present and it has its own replicating machinery, and it has its own replication machinery. And we also know that plasmid is a very extremely important tool, I would say, which is used in cloning. That it acts as a vector in transferring genetic material from one organism to the another. So the role of plasmid seems very easy, that it's extra chromosomal. It provides extra genetic material to the DNA and to the cell, and also it is capable of transferring genetic material from one organism to another. The cell wall. The cell wall, as we know, it is the protective protective barrier of the contents of the inner contents of the cell. It is mostly it functions in protecting and maintaining the structural integrity and being extremely permeable and selective to the entry of foreign agents or nutrients within the cell, as well as permeable to the exit of nutrients. So the cell wall plays a major role in protecting the cell contents. It shouldn't, like, all the important things shouldn't come out, or some foreign material which could turn out to be toxic for the cell shouldn't. Now coming to fimbriae and sex piles. So this is something which seems to be slightly confusing when we discuss, because most books synonymously use the term fimbriae and sex piles. So a pili, pilus, or a fimbriae and fimbriae, these are kind of appendages that are present in the outer surface of the bacterial cell or of the cell. You can see that you cannot distinguish between them, that which is a fimbriae and which is a pilus. Same goes for here. So is a fimbriae is a sex pilus and a fimbriae. How are they different? So a sex pilus is specifically used for conjugation purpose, transformation purpose, that is for genetic transfer. Also, in certain cases, it helps in motility. Whereas fimri are mostly uh, used fimri attachment. It can attach, or it is mostly used for motility purpose. Also, it helps in motility and attachment. So now, then how does the sex pilus fimri make it different from flagella? Now, bacterial flagella is exclusively used for motility purpose, and it is a locomotor appendage. And also, what happens is it serves as a virulence factors. Like many bacteria have are virulent. We know they are disease causing, and the fibri is the reason why it acts as an antigen within the body. That is how it is recognized by the immune system as an antigen. So that is the major difference between the pilus and flagella and the fimbriae. So both of them are slightly hair-like. They have a thread-like and a hair-like appendage. They cannot be visually seen with naked eye. They need to be stained. There are very specific staining things, staining techniques that we use for fimbriae, and yes, very specific staining techniques we used for observing this, and it's very easily observable under the microscope. So yes, I repeat, bacterial fimbriae helps to helps in motility, as well as it facilitates the bacteria to adhere to certain substrates or substances, like in rocks, in streams, or even in host tissue. So it also is one of the serological factors. And sex pilus is used for conjugation, transformation, mostly the genetic transfer between two organisms. And the flagella is an exclusively locomotor appendage, and also it acts as a virulence factors for some cases, and to a certain extent this can help itself attach. So by locomotion, it's not just movement. To a certain extent, if we see locomotion and attachment to a substratum or a substrate, or adherence seems to be synonymous. So yes, it locomotes as well as helps it to attach to a host or to a particular surface. So in this particular picture, if we just zoom in and see that this is a bacterial cell and this is one more bacterial cell, and this something is being connected between two. So this is the sex pilus, and through this the extra chromosomal DNA is transferred into it, and genetic transfer happens. So a bit more detailed. So this is the sex pilus, a bit more in a closer view, and these thicker ones are mostly flagella. Flagella thread-like and thicker with respect to fimbriae and pilus. So yes, so that's all. So if we now try to match the following of this question, what the it was initially was. So the nucleus is the genetic material. It will be one of two. Plasmid is the extra chromosomal DNA, 2, 4. Then fimbriae, it facilitates adherence to the substrate, 3, 5. So till now, it will be option C. Yes. And cell wall, it protects, and 5 of 3. So yeah, it was a pretty straight question, but yes, this is a very something which confuses us most of the time, that what is the difference between pilus, fimbriae, and flagella. So I guess I'm able to clear this up. Okay. All right. Then let's move on to the uh, next question. Yes. Okay. Now, let's move a bit to a different topic about pedigrees. Now, this is a question that was given, and we were asked to find that what type of genetically linked disorder it could be observed, like seeing into this. So before getting into what kind of it was observed, we can just do something that let's just discuss how a pedigree is done and what are the different aspects of pedigree. Okay. I'll share one more screen with you so that it's a bit easy. Share. Hmm. I hope my second screen is visible. Just reply in the chat box. Okay. So, hmm. So this is how this pedigree looks like. Let's discuss what a pedigree is. So it is actually a chart which is represented in the form of tree. Now, why do we say it is a representation in the form of a tree? We have a chart. Yes, visible. Okay, thank you, Amardeep, so much. Okay. So why tree? When we say a tree, it means a family tree. Like pedigree is actually a display or a visual representation of a family that could possibly have a disease, and it could suppose a disease is being transmitted with the family, and we need to know what is the order of transmittance. Like, is it coming from the father, mother? It is an X-linked, Y-linked, or maybe an autosomal recessive, so that the future generations, if possible, we are able to know that yes, this the source of this gene or the particular disease could be this, and then there are ways of prevention too. So that is how a pedigree helps us in predicting that what the future generation will look like, or what will be the genetic trait of the future generation. So yes, if we see in the middle, this is I just have given an example. So it's a family uh, in which one now how is it before going to that, how is it represented? So the circles are females, squares are males. And if the circle or square is filled with some color, which is black generally, or it could be any color, that means that particular individual is affected. Now, these mean one, two, three, four. These are the generations. Like this is the first generation, which is which has the grandparents, then the parents, their children, and then their children. So this is how this 1, 2, 3, 4 mean that these are the generations, and these indicate this 1, 2, then 1, 2, that which individual of which generation, like individual female 1 of first generation. Similarly, this is female 2 of second generation. So this is how we try to locate, like if we have to annotate that, okay, where do they lie? Now, these are the basics. Okay. So the founding parents are mostly written in the top. This one. These are the founding parents. And then eventually there's their children, then they get married. Now, these straight lines, yes, these straight lines indicate that they are linked. Like they have mated and they have a child, or they have an offspring. These and these horizontal lines and these vertical lines indicate that how many children do they have. Here, generation 1 has 1, 2, and 3 children. Now, if these two are linked, it means that they have these 2 children. Now this square is not linked to anything, and it is completely unlinked, and it is showing it is mated with this one. That means this particular individual in black, that is 2, 2, got married to this person, and they have now 3 children. So these are the basic things how a pedigree looks like. Okay. So now, whenever a pedigree you get a pedigree, or you are asked to solve a pedigree, the first thing that should be that you need to see that a particular disease. Now you'll find that mostly it's in dark. Some members are in dark, some are in light. So you'll have to find that what is the mode of transmission of the disease. So what happens is you need to see there are 2 factors. One is the dominance, that whether the diseased allele is dominant or it is recessive. Like just like in Mendelian's inheritance pattern that we discussed, that dominance means that if a particular trait, suppose it's capital A and it's dominant, okay, so A alone is capable of creating the disease or leading to the disease. So if it's AA, it's a lethal thing. It will definitely be diseased. But even it's A and A, still it will be disease. So that is the concept of dominance, that one single allele is capable of showing the phenotypic effects of the particular trait. Now, this is dominance. And second is recessive. Recessive means that if a particular trait is A and A, only then it will be diseased, because it needs two of the same alleles to produce one disease. And A is alone is not capable of no disease. Okay. Yes. Clear? Now, there is one more thing. Once you know it's a dominant or a recessive one, that's fine. Now, the second is the linkage. That is it coming from the X, that is the sex chromosomes, or is it coming from the autosomal chromosomes? That now we know that there are two types of things. One is sex, X-linked, and one is autosomal. Autosomal means the somatic ones, and X means the X and Y concept. That X means from it will come from the mother, or mother will be a carrier, and in that way, like it depends on the gender of the parent, or it depends on like the normally the somatic cells, like living from the genetic gender of the parent. So these are the two things that are very important, that one is dominance and one is linkage, like what is exactly happening. So we need to know. Now, like I'll be discussing very basic things once again. One is the autosomal chromosomes, that we know we have 23 pairs of chromosomes. So out of that, 22 chromosomes pairs are autosomal, and the rest one is XX or XY. If it is XX, it's female, and if it's XY, it's male, and that is the sex chromosome. And there's a condition called hemizygous condition. So males are hemizygous. That means that not completely zygous. Hemi means same. So a male is always XY and a female is always XX. So it is hemizygous for X. So that is why X-linked gene. So males have only one chromosome, which is X, and correspondingly one allele for any X-linked gene. So anything that has X-linked will be one allele. But and allele, as we know, it is the variation or the version of a gene which is present in multiples. It could be 2, it could be 3. We have discussed in the last session. So in terms of sex chromosomes, females have 2 alleles of X-linked genes and male have just 1. So that makes up X and Y and X and X. So I guess it's clear what I have a chat. Okay. Yes, clear. Now, before solving a pedigree, there are some basic assumptions that we made. Now we know that, okay, I have to look into it's autosomal, it's X-linked, or it's dominant or recessive. Apart from that, there are certain things that we have to keep in mind. So let's think one by one. Okay. So just a second. So the general assumptions is that is it controlled by one gene with 2 alleles, a dominant allele or a recessive allele? Okay. Why one gene? Because again, the concept of if it's X gene, then male will have just one. So it's just one allele, and it's a dominant or recessive. So that is always that concept will always be there. Okay. So there are also 3 things that we generally consider. The concept of complete penetrance. So what is complete penetrance? So is that when an individual in a pedigree will be affected, that is, it will express the phenotype associated with that affected trait when the individual carries at least one dominant allele of the dominant trait or 2 recessive alleles of the recessive. Just what I mentioned in the previous slide, that what do we mean by this capital A, capital A, capital A, small a, small a, small a, and just A. That what actually happens, that is called complete penetrance. Now, rare in population, so in each problem or in each sum that you're given, the trait in question is rare in the general population. So these are rare diseases. It's not like it happens rampantly among everybody. So we have to assume that the individuals who marry into the pedigree in the second or third generations are not carriers. Most of the time, we have to think that, okay, the ones that are coming from outside, that you have a pedigree, and some you will find second and third generations that they get married, they're getting married, the children, the offsprings of the first generation are getting married to people outside. So we have to initially assume that, okay, these are unaffected and they're also not carriers also. So it's a very presumed and preliminary thing. It's not the final thing. With the presumption we start, and as soon as we start unearthing or unfolding the pedigree, we see that, okay, they could be carriers, they could be affected individuals only, and that we did not know, and that is the complete entire purpose of a pedigree analysis. So this does not apply to founding parents, that, okay, so either of the individuals or the term could be carriers. Like the founding parents could, of course, be carriers. It's not the people marrying outside, those individuals cannot be carriers. We'll consider that, but parents could be carriers, the main founding population. So founding parents are the ones which are placed at the top, the main parents, the grandparents that I mentioned. Okay. Uh, generally, not always, we don't consider it to be violent, because if it is violent, then again it will go from like father to son, son to father, like that. Like it will not violent. There's some limitations about violent. So it is like it goes exclusively from father to son. So generally we don't consider. But again, if a pedigree keeps on showing that every generation has a father and then a son affected and again a son affected, like if I arbitrarily draw, suppose it's a father, it's a mother, I'm not sure about the mother, then they have another son. Uh, yes. So again, suppose they have three children, son, again a daughter, and the son is affected, father is affected. Again, from here, he marries to someone like this, and again the son is affected. So that gives us an idea, okay, this could be like father to son, violin, but generally it is not. So there are 5 clues. Like if I would suggest that go back and just see that what these clues mean. So I'll be discussing now. So there are 5 clues that we have to keep in mind before solving any pedigree. One is an unaffected individual cannot have any alleles of a dominant trait. So in order to have a dominant trait, it has to have A, or AA. So if somebody, or maybe for a dominance, I'm saying. So if somebody is unaffected, he or she cannot have A, this capital A in it, because one A is equally capable of creating the disease. So it cannot have. This is one. Individuals marrying into families are assumed to have no disease alleles. That they will into the family, yes, they will never be affected and can be carriers of recessive, because trait is rare in population, that we discussed, that rare in population genes, that any kind of this diseases are kind of rare. So it's considered that because this trait is rare, so they cannot be carriers. Third is an unaffected individual can be a carrier, have one allele that is a recessive. Now, the trick of recessive is recessive could be like, for a recessive trait to be a disease, it should be AA for disease. But to be a carrier, it could be just A. So anything in small, it could anything recessive, just one allele is capable of making that individual a carrier. That person will not be affected, will have no phenotype, will not. But if it transfers to its sibling, its offspring, and that offspring gets another allele, which is again a recessive one from another parent, then both will join together and form the diseased one. So yes, an unaffected individual can definitely be a carrier, have one allele of a recessive trait, because 2 alleles of a recessive trait are required for an individual to be affected. So if 2 is not there, half of it is there, it is a carrier. That isn't one more assumption. Fourth assumption is when a trait is X-linked. Suppose a trait is X-linked. A single recessive allele is sufficient for a male to be affected. Okay, because males are hemizygous and it has only one allele of the X. So if one X is present, the male is itself affected. Okay, if it is a link, like a X linked thing. Uh, yes. But if it is a X linked dominant one, then this has to be XA. X, if it's XAY, then the male is not affected. And if it is a recessive one, then this alone is capable. So just I'll write it here. If this is a recessive one, it will be a diseased. But if this is a dominant one, it is non-diseased. Okay. So that's the catch. Hmm. That's the fourth trait. And what is the fifth one? A father transmits his allele of X-linked genes to his daughter, but not his son. So it generally happens in a crisscross way, that father to daughter, mother to son, and again like that. It will never be to his son, because if it is to his son, that as I mentioned, it will be a father to son Y-linked. It's a Y-linked disease, but an X-linked disease will always be a CRISPR. So if I just write it here, it will be father to son, um, daughter, sorry, mother to son, like this. It cannot be in the opposite way. Okay. So that is what is written here. A father transmits his allele of X-linked to his daughter, but not his sons. And a mother transmits an allele of X-linked genes to both daughter and son. Okay, yes. So in terms, if it's a disease thing, mostly it's in that from mother to son, it is transferred, the disease is transferred, and into its daughter, the carrier form is transferred. Because why? Mother is, if the mother is not affected as a carrier and the father is also a carrier, the daughter has a high probability of getting the disease, because both are X and X and X linked. But the son will definitely get the disease in this case. So keep this in mind. A father will transmit his X allele to daughter, but never to the son. A mother will also transmit the X-linked disease to both daughters and sons, but the probability of disease going to the daughter or the son is different here. Okay, let's talk about some like more. Now let's get into the patterns that what are exactly happening and what is happening. Okay, so if we see here the pattern one, this is one, maybe two, and then is three. Okay, so in pattern, what do we see here? Here is a female, here's a male, and this is a affected female. Okay, so what is it? Like a particular female is affected, but its parents are not affected. So what could have actually caused this female to be affected? It's a recessive one. What did I say? To be affected, if it's a recessive one, then its parents can also, it could be the parents are not affected. Why? Because to be affected, it has to be A and A. So the mother was heterozygous in this case, mother and father both. And what happened is it transferred both of its A and A to this individual, and that became diseased. Okay. So this is one case of recessiveness. That if any affected individual has two unaffected parents, in such a genetic disease, at least one parent must have the allele for the disease. Okay. Also, now, if neither of the parents are affected, the trait cannot be dominant. So yes, we have already that none of the if neither is affected, the trait cannot be dominant. So it's a recessive. So this is how a recessive thing is happening here. Now, even in case of, you see, this is a this is a male that was affected. First we discussed a female, and this was a male that was affected. Affected AM, that is an affected male. Same thing is happening, that at least one recessive trait was there, and both of one parent, at least one parent has had the, both of the parent had one allele, and that was transferred. Okay. Now let's see more things. So autosomal recessive is like if affecting founder daughter has two unaffected parents, the disease must be autosomal recessive, that we discussed here in this scenario. Affected individual must inherit the recessive allele from both the parents. So both parents must have an allele that both of them had this small a. So it got this A and A. Now, if the father had a recessive X-linked allele, he would have to be affected since he has only one X-link. Now, if this was an X-linked concept, if the father had, suppose this was an X-linked thing, but the father had to be affected because father has only the X and Y, and that X comes from an affected mother. But no, that is not happening. So here he is unaffected. So this is like a example of recessive. Now, in order to be recessive, if an affecting founder son, now let's come to the son example of the son. Now the son is affected, it again has two unaffected parents. So we cannot exactly determine that the recessive disease is autosomal or X-linked, because what did I say? From mother to son, that transmission occurs. So now if then the mother could be a carrier, that she herself doesn't have the disease, but she's transmitting the carrier gene to him, and he is getting affected by the other gene that is recessive, which is coming. Like suppose affected male, so it would be like this. And Y will be coming from here, and XA is like the carrier form in the mother, and so that is happening. Okay, so that is how affected male. So this could be like that. But if the trait is autosomal, both parents can be unaffected carriers of the disease. If the trait is X-linked, the son must have inherited its allele from mother only, as his father cannot be affected. Just I mentioned that mother was carrier, she transmitted it to the son, but from father this cannot happen. So this could be an example of X-linked recessive disease also, or in general it is a recessive. Now let's get into more details of the X-linked recessive one. So when an affected non-founding son, okay, this one has two unaffected parents, the disease must be X-linked. So what does that mean? That non-founding son, this one, had two unaffected parents, and the disease must be X-linked. So the father who is marrying in doesn't have any disease allele since he's marrying into the family. So this one, so the affected son, this son had inherited this allele only from his unaffected mother, which was again given by the affected father. That is how it came to him. So the affected son inherits allele from his unaffected mother. A male cannot be affected by a single autosomal recessive allele, but can be affected by a single X-linked recessive allele. Is it clear? Also, a male cannot be affected by if it is a single autosomal recessive allele, it has to be two to be affected. But in case of single X-linked recessive allele, it can be, because the disease trait is X, and if this goes to the father or the male, it will be diseased. It will have a disease. Clear? So I guess it's briefly clear that what the other different options we get, or the different assumptions we have in case of autosomal recessive. Okay. Now this one. Now see what happens in case of a dominant. So in dominant, a very clear pattern. Okay, I just go back to here and you see in this example, you see there's a generation skip also. That one generation is affected, other generation is not affected, and again the next generation is affected. This is also a very important visual trait, a visual pattern that we need to remember while doing this. If in case of recessive diseases, there will be a generation skip. Because why? Unaffected will have heterozygous condition and affected will have homozygous conditions. And in one generation, this will be alternating. So there are high possibilities that there will be a generation skip. Now, let's take up from this point of generation skip in case of the dominant trait. So you see these both examples, what is happening, that there is no generation skip. All generations are affected, right? Here it's the same, in one male is affected and one is female affected. So now let's see. So a disease must be dominant if every affected child of non-founding parent has an affecting parent. One parent has to be affected to transmit the disease to the next generation, irrespective of the fact that the other partner was affected or not. So one parent minimum has to be affected. Okay, affected to transmit it to the next generation, irrespective of the fact that the other parent was diseased or not. Why? Even in heterozygous condition, it will be diseased, irrespective of the fact it's male or female. So this will be in general a dominant trait. So an unaffected mother, suppose an unaffected mother who is marrying in does not carry an allele for the disease. So the affected child inherits the allele from the affected father. So if I try to draw this, okay, and it could be a son or a daughter, anything. This is not. So the mother was not affected. The father was affected. The mother might have a recessive allele. She didn't have, she was not affected means she had a recessive allele, but the father had maybe a heterozygous allele. So some way or the other, this child will have a, be it a boy or a girl, will always be affected. Okay. So the affected child inherits some allele from the affected father. No child could be affected by a single autosomal recessive allele or X-linked recessive allele, so the trait is dominant. So again, there it's a repetition of the fact that no such allele could X-linked recessive allele could be transmitted. No child could be affected because why? It has to be two in number, not one, to be affected. Uh, Single allele is not capable of causing a disease, be it X or recessive. Okay. Now the last one is the, let's get into more discussion what is happening. So when the affected son, the first of non-founding parents has an affected father, the disease must be autosomal dominant. Okay. Now we have to see that it's dominant. Now it's autosomal or X-linked. That is something we need to see. So if the affected son of the non-founding parents, this one, non-founding parents has an affected father, disease must be autosomal dominant. Why? The father will not transmit any X-linked allele to the son, so the disease cannot be X-linked dominant. Am I clear here? That this person is affected by the father, but that is not because of X. Because father does not transmit this X-linked allele to the son. It transmits it to the daughter. To the son, it cannot give that, right? So the disease is not from the father, like not from the X of the father. It is normal from the autosomal origin. Again, if a daughter of the non-founding parent has an affected father, it is again difficult to determine that the dominant disease is of autosomal or of X-linked. Why? Because the father is capable of transmitting either the autosomal dominant allele or the X allele, which is dominant allele to the daughter. Okay. So this person is capable of transmitting both if it is of XA as well as XA to it. Autosomal dominant allele, it could be, or a X-linked dominant. It could be A or XA, both. He could transmit both of them. So here determine. Okay. Thank you for joining back. Uh, Thank you for joining back. So we were discussing that if a female is affected by a affected male, and again it has a non-founding parent, so it is slightly difficult to determine that whether that particular trait was dominant or that particular dominant disease is autosomal in nature or an X-linked disease. Why? Because the affected father can transmit either of the it could transmit both the autosomal dominant allele, that is capital A, or an X-linked dominant allele to the daughter. So it's a bit difficult there. Now, coming to the question. This is a bit more detailed things, that what are the different indications, like I've already discussed that this is the male, this is the female, this is mating, parents with one boy and one girl in order. And keep in mind that whatever comes first is the order, and which it is important because we know that the when we are solving that probability of having two sons in a row, maybe one son and one daughter in a row, those probability calculations, the order is very important. Then if they're twins, we mention it like that. If they're monozygotic, the sex is unspecified, then number of children's like that affected, death, and these are like more things. And if we do a consanguineous marriage, that is within blood relatives, then this one many a times we'll have this also. Rest things are less common. So now if you are given a pedigree like this, that is the question, what should be your first thing, that while seeing this, what is my first observation, that uh, sorry. Yes. So, what is your first observation? Okay. D. So, why is it D? What makes it X-linked dominant? What happens in an X-linked dominant disease that it makes it this things? Okay, I will discuss that rather than confusing you. So, also one more thing, if it is a X-linked dominant disease, uh, more females are affected. This is one more assumption that we are, it's not mentioned there, but more females are affected. Why? Because 1XA is capable, like 1XA, and is capable of causing the disease. It's just like the same, like if it's XA coming from a mother and also in a heterozygous condition, also it will cause the disease. And in case of X-linked recessive disease, more males are affected because one X small a is capable of causing disease to males. So more males are affected. In this example, two things I have observed, that is none of the generations, there's not even single generation where the disease has not taken place. So the disease is present in all generations. Okay. So that makes it a dominant one. And also if we see, the ratio of females are much affected, females are much more greater than males. That this one, this one, this one. Okay. Females are extremely affected. So yes, this is an X-linked dominant disease. No, Mardit, this cannot be X-linked recessive. What I mentioned, that if to be a X-linked recessive one, it has to, like, more males have to be affected throughout the population, throughout the pedigree tree. But you see, females are more and more prone to it, and they have more effect. So this is an X-linked dominant disease and not a X-linked recessive disease. Is it clear? Hmm. Yes. Okay. Now one more question. Now let's see that what this says. Uh, Uma marries Saket. Both has normal vision. However, Uma's mother is colorblind. What is the probability of their children to be colorblind? So these are the four options. Let me just try to draw the pedigree out of it and see let's what happens. Okay. So Uma is a female. I'll draw it like this. And Saket is a male. They get married and we have to find the progeny. Suppose I have four of them. Okay. In what proportions are they going to be colorblind? Why? because it is known that Uma's mother is colorblind. So this is Uma's father. This is Uma's mother, and colorblind is a X-linked recessive disease. So it will be like yes. Oh, 1 minute. Hmm. Hmm. So we'll be discussing that. What is it all about? Just give me 1 minute. Hmm. So colorblind is a X-linked recessive disease. So what happens in case of colorblind is that it has to have both XA to be a diseased one. XA is capable in case of males, and for females it has to be both XA and XA. So yes. Hmm. And so this XA was transmitted to Uma, and she became a carrier here. And he was unaffected male, and also unaffected male. So we need to find how many children will be affected here. So before getting into that, let's just discuss different like what happens. It actually happens in the colorblind scenario. Okay. So suppose there's a parent without a hmm, hmm. So there's sorry for the delay. So there's a parent without the gene who doesn't have that recessive particular gene X and Y, and this is a male parent. Okay, and there's a carrier mother who does not express the trait. So what I said that about carriers. See, okay, we'll discuss. Archana will discuss that what is the scenario. Okay. So carrier mothers are what are carrier mothers who does not express the trait but just carries it, has no phenotypic expression but carries it. So he's a mother. So if we see in the genetic makeup, it's XY, and this gene is actually responsible for the disease and that's been carried on. So this is a female child, this is a male, this is a female, and this is again a male. So if we see here, number, so who are going to be affected? And minded, it's a recessive one, recessive. So a female, in order to be affected, should have X and X, and a male to be affected, one X is just capable. Okay. So any male having small x and y is diseased. So this male is diseased, this male is not diseased, and now the females are diseased. Again, this female is a carrier. So is it okay? The daughter, so that is what is here. Daughter who is a carrier but does not express the trait. Daughter and son who do not carry the gene nor do express the trait. This one and this one. XY and XY. Okay. Son who has the gene and expression of the trait or the condition. This one. So this is the diseased one. Oh, this is a female. Sorry, like this is a female. Okay, this is a female. This one is a male. This one, and this male is the one who is affected. Now, let's see the different combinations. What happen when a colorblind man and a non-colorblind woman act marry? So we see that there's a colorblind man and a non-colorblind woman. So colorblind man means X is he's a affected colorblind man, and a mother who is non-colorblind, and neither she's a carrier nor a affected person. So just if we cross and see, wherever this small x will be along with a y will be affected. So none of the sons will be affected because y will not be coming from mother, no, from father it will come. So none of the sons are affected. But yes, if that man transmits that X carrier, like the X gene to any of the daughters, all the daughters will have 100% chance of being carriers, not affected. 100% chance of being colorblind carriers. And when they again mate with someone, and this same scenario will happen. So what scenario will happen? Now, suppose this was the daughter who mated with a non-colorblind man, non-affected, non-colorblind man. This one and this one. See what will happen. This is small x, this is the x, and this is the y. 50% of the sons will be colorblind. By 50%, that means that if he has two sons, one out of two will be colorblind. Okay. And in case of females, what will happen? Only 50% will be carrier. One out of two will be female carriers. Okay. These were the two examples that what happens in. And again, let's see more two examples that now a colorblind man and both a colorblind man and a colorblind woman marry. Now the children that they have, 50% of the sons will definitely be colorblind. Why? Because either of the parent is will supply the X gene, that is affected gene, and the son will have it from either of the parent. It could be from the mother or it could be also from the father. So definitely the sons, 50% their chance that they will be, and one might escape and will not be a colorblind. And in case of female, this is very important, one female, that 50% of them will be affected. Now we have affected females also. Okay, apart from affected male, we have affected female also, and that is also one out of two, that is 50%, and the other one will be a carrier. And similarly, if a colorblind mother marries a non-colorblind father, what will happen is all the sons will be colorblind. Now the mother, all the X genes that will be coming from mother to son, both the alleles are colorblind. That both XA, like X is affected gene. So whatever the number of sons are, the two sons, both of them will be colorblind. And in case of the daughters, they will be carriers because this particular gene will make it a carrier and not a because why the egg that will come from the father is not affected or not a carrier, so they will be carriers and not like affected. So are these examples clear to all of you? Hmm. Okay. You can just stop me because these are slightly complicated when you're studying it for the first time. I suppose you're not studying it for the first time, but you can stop me and we can discuss once again. So this is a family tree. You can just go back after we're done with the class. I'll not explain this in detail. So this is a real family scenario in which this pedigree was done, and this is how it turned out. So now coming to the question, now let's see what actually happened. A lot of back contextual story. So I'll just draw it once again, or maybe I'll go back and draw it here itself. Yes. So, an unaffected male, unaffected, yes. Uh, Yes. Okay. So this was Uma. Yeah, this was Uma. Uma's mother and father, and this was Uma. So Uma was a carrier, and Uma will be a carrier because one XA will be coming from Uma's mother, and that will make her a carrier. And the father is unaffected, so the person who is marrying Uma is unaffected. So the different progenies that will be formed is like this: that XAA, XA, then there will be a XA, Y, this and this, and there will be again a X, capital A and Y. So this male is affected male, and this is non-affected male, and this is again a carrier and non-affected. Okay. Now, if we see, out of two sun, one is affected. So 50% sun is affected, and one of them and 50% is carrier. So now let's just go back to and see what were the options that we had. Yes. So all male progeny, colorblind, phenotype, all female non. No, this is not that. Half of the female progeny colorblind. No, half of them will be carriers. Sure shot. And all progeny will be normal. No. Then half of the male progeny will be colorblind. One of them, that is the one which will have the X, A, and Y, is the colorblind male. Okay. So that's how we solve it. This is solved. Okay. Let's go to the next question. Now, so we need to now match the following of the different chromosomal disorders with the correct genotypes. Now, these are examples of aneuploidy where the entire set is missing or there's problem with the entire set. So I have added a table, this one, for your reference so that it's easy for you to do the match the following. So see that in case of normal chromosome, we have four sets. We are diploid organ. We have two pairs of each genes. So this is the normal set. And in case of different aneuploid condition, nullisomic, monosomic, doubly monosomic, trisomic, tetrasomic, or doubly tetrasomic, you see that if it is affected, it's affecting the entire set. Like in nullisomic, one entire, this two of these chromosomes are not there, right? Here, one is missing. Doubly monosomic, it is from two sets, one is missing. And here we have one extra. That apart from the 2n, we have one more extra. We have two extra in tetra. And in doubly tetra, we have four extra. Like here 4 and here 4. Instead of 2, it's 4 and 4. So this is where these problems lie. And as a result, they directly affect the individuals, which leads to Down syndrome, Patau syndrome, Edwards, Turner syndrome, these different kinds of syndromes. So if you look into this table, trisomic 21 leads to Down syndrome. Like trisomic 21 means you have one less, right? And Patau gives trisomic 13 and Edwards gives 80. And similarly, monosomic Turner syndrome is X, so that one X is missing in. And in case of trisomic clean factor, it's like, normal. It is, or trisomic, it's two X's and one extra Y. So a particular male will develop some female characters also in it. So that is what happens. And if we try to match the following, it's like Down's syndrome, it's one of two, that is trisomy of 21st chromosome, that the 21st chromosome is affected in that way. Then Patau is trisomy of the 13th chromosome. And these all affect the particular positions of the chromosome, particular chromosome numbers. Okay. So that is what happening. And Turner's is 4th, that is monosomy. And Edward is trisomy of the 18th. So this is clear here. It's clearly like what is happening, that it is affecting the chromosome set. It's 2n minus 2 or 1 or 2n minus 1 and 1. Like 2n minus 2 means the entire set is missing. 1 means 1 from the one set. 1 minus 1 means 1 from each set. Plus 1 means addition of 1 more to already present set plus 2. And so if you see, these have decrease in number of chromosomes and these have the increase. So in that way also you can remember that nulli, mono, doubly, tri, tetra, double tetra. So I guess this is clear. Yeah, see. Okay, let's go to the next question. Now we'll shift slightly from Mendelian genetics and like clear minds and clear our minds out of it. It's a lot of that. So here this question is that a student is trying to learn gram staining. So gram staining. So what is gram staining? And he has written the protocol in a random order. He was mentioned, okay, you have to do this and this, but he has somehow like misplaced and his order of okay. Okay. The protocol order has just gone random and he is now very confused how to do. So these are the four things. Now, before getting into the question, let's discuss what is gram staining. Okay. So, gram staining, what is gram staining? Gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particular color. So, gram staining is a process of staining a cell with a particu Gram staining is a process of differential staining in order to distinguish morphology of two different bacteria through a same process, or two different organisms through a same process. So this gram staining process has different steps. Any anything that you want to stain, that you want to color it and see under the microscope, generally it can be not be done with live cells. So live cell has a different total aspect of observing it under the microscope. But whenever we're staining it with these kind of basic or acidic dyes, so basic and acidic dyes depend on the color of the dye, whatever it has. Okay, so first step is fixation. So the term fixation means in general, in literal meaning, if we go, fixation means to fix it, like to fix it to a surface so that all the morphology is intact, but we are able to see whatever was there in the live condition, that is also observable under the microscope. So we need to fix a snap freeze, that we need a snapshot or a snap view of what is happening in the live cell, right? So for that we fix it. Now generally, the after fixing it, the internal external structures are preserved and fixed in a particular position. But what happens is it inactivates the enzymes, and that has a possibility of disrupting the cell morphology and toughens structures or change during staining. Yes. So the morphology is slightly disrupted, but that will in turn toughen the structures and they don't change when staining is done. So all the properties are intact, but its morphologies to a certain extent gets disrupted. So, and fixing processes generally involves killing the cells. So definitely they'll be killed and they'll be attached to the slides. So there are two different processes of fixing. One is heat fixing and one is chemical fixing. So in heat fixing, we like pour 1 ml of maybe a solution having the cells, make a smear with the help of an inoculating loop over a slide. So it's something like this, that if this is your slide, you just put small drop of sample which is a solution which has the bacterial cell on it, and you just smear it like this. Okay, you let it dry and you pass this in a pass this over a flame in a Bunsen burner. You pass this over a flame. So the film of cells, which is a screen, are generally heated, and that heating will let it get it fixed. So it will preserve the morphology but inactivates all the enzymes. And however, it will destroy the proteins in the subcellular structure, which may distort their appearance. So heating is not a very optimized or optimum technique that we use. Generally, while doing experiments in the lab, we use it, but for better precision, we use chemical fixatives. So what are chemical fixatives? So chemical fixatives will protect all the fine cellular structures. It will not distort anything or the morphology. So there are different chemicals which interact with the organic compounds or the compounds which are present in the outside of the cell, and that will make it attached to the cell. So commonly used are like glutaraldehyde, formaldehyde. Formaldehyde, the CHO group acts with the CHO group of the cell, and that is how it helps to fix it over the surface. So once you know that your sample is not moving and it is fixed, now you can continue with the staining process. Okay, so what I was discussing, that differential staining. So a differential staining is a process that distinguishes organisms based on the staining properties. Like what is suppose I want a gram positive or a gram negative bacteria. So if I want to distinguish it from the same experiment, that is where differential staining comes. And what do we use? There's one more thing. Now, coming to gram positive and gram negative bacteria, you know the morphology of gram positive and gram negative bacteria is slightly different. Like the peptidoglycans makeup of it is different. So a gram positive bacteria has a thicker peptidoglycan layer. The cell wall is slightly thick, and whereas in a gram negative, the cell wall is thinner. But the space between the plasma membrane and peptide diagonal there is also small in case of gram positive, and reverse, that is thick. And one more thing, sorry, this is not peptide. This very important thing is the outer membrane. That is, an extra membrane is present in case of gram negative bacteria, which actually makes it different in the staining process from the gram positive. So this outer membrane has teichoic acid, which is negatively in charge, and it's very helpful and useful. We'll see that how it happens. Now, whenever we are saying we are staining a microorganism, suppose that stain is alone is not capable of binding to the subcellular structures. So for that, what do? We supply a mordant. So a mordant is actually a salt in nature which binds to the dye and forms a complex, a coordination complex, and that enhances the binding of this complex to the subcellular structure that we are targeting. So here, that is why we use a mordant. So here we use iodine. So iodine interaction, like, increases the interaction between the cell and the dye so that the cell can be stained more strongly. So now look, let's, idea we have, the idea that what a gram staining is. Now let's look what happens. So the left panel is different kind of bacteria, like it could be rod shaped, it could be cocci. So what is the first thing is that we stain the cells with, no, teichoic acid is present in gram. Are you asking me that Amardeep, teichoic acid is present in gram? Yes, yes, sorry. Teichoic acid is present in gram positive, and the outer membrane has this lipid layer which gives, it imparts a negative charge. Sorry, thank you for correcting this to me. Yes. So teichoic acid is present in gram positive, whereas the outer membrane of the gram negative cells have this LPS layer, the outer polysaccharide, lipid polysaccharide layer, which gives us the negative charge. Yes, you correct. You're correct. Thank you for correcting me. So now let's look into the steps of staining. So what happens is, first we provide stain the cells with crystal violet, which will give this all the cells, irrespective that it is a positive, it's a gram positive or a gram negative cell. So it will color it in purple, like purplish a violet color kind of that. So we see that all cells are now purple color. Okay. So now then what happens is we rinse it. Whatever color has to bind, it will bind, and then we rinse it. Then we apply the mordant to it, which is iodine. And what happens? Again, we rinse it. Now, this iodine will enhance the binding of the gram stain to the cell. Okay. So done. The next process is the decolorizing agent. So decolorizing agent is an alcohol we use. Different concentrations can be used. We use an alcohol. So we wash the cells with a alcohol for like 10 to 30 seconds under water. So what will happen is, gram positive cells will have the purple color, but the gram negative cells will start losing its purple color. Why does now this happen? Because gram positive cells have that thick peptidoglycan layer. So the gram stain has got inside, but it will not get out easily. And again, even after using a decolorizing agent, what happens is, the pores within the peptidoglycan layer are shrunk, and no color can come out. But in case of gram negative, the peptidoglycan layer itself was very thin. So what happened is, the color went in and it easily came out after using the decolorizing agent. So it decolorized the entire cells. Now, on we put one more color on it, one more stain on it, which is known as the counter stain. So we counter the first color with the second one, which is saffron, and it gives a pinkish hue or a pinkish color to it. So what will happen is, gram positive cells already have the purple color. It will not be affected by it. But the gram negative cell, which is lost is purple color, will now be stained as slightly pink. So we will see a combination of pink and purple cells. So the purple ones are gram positive and the pink ones are gram negative. Clear that why they take up this stain and why not? What is the difference? And it's in that logic that gram positive takes up gram stain, so it's gram positive, and negative cells cannot take up the gram negative. So that is why it's gram negative. Is it clear to all of you? Is it clear to all of you? Okay. Hmm. Uh, so let's see. So what should be the order of it? And now just now it's quite clear. So what should be that? Just some one of you try it. Do I have answers for it? No, not C. Any one of you just try to solve it. A. Yes, it's A. So first is we heat sterilize the slide. Heat sterilization can be to just kill the to make it aseptic and to kill all the unwanted organisms. One, and second is like to fix. Here fixing is not written, but it could be fixing also. The step is because of fixing. Second is we subject it to crystal violet staining. Third is then grams iodine is added for addition of affirmation. Then five. Then comes the decolorization step and followed by saffron staining and viewing under the microscope. So yes, this is how this is this these are the different steps of gram stain. Yes. Is gram staining clear to all of you? Okay. Now, why did this teichoic acid and why did I mention about the like the outer lipopolysaccharide layer and the teichoic acid. Now, when we discuss about acidic stains and basic stains, it happens on the basis of color, like basis on the charge. So, some subcellular structures which are negatively charged will bind to positively charged stains, and the ones which are and the vice versa. So, bacterial cell has negative charge. So, like the negative charge comes from two sources. One is the teichoic acid and gram positive. Both the bacterial cells, be it positive or negative, and gram negative cells have the outer layer. So that is how it has negative charge and it binds to positively charged stains. It positively charged acidic stains. And similarly, the other nuclei, cell wall, and plasma membrane, all of these are negatively charged. So they will bind to acidic stains. And basic stains, which are positively charged, bind to negative components of the bacterial surface. Uh, so that's a concept that how positive and negative stains or acidic basic stains, positive negative charge is coming. Okay. Now let's discuss a different kind of question. So there's an antibiotic has been applied to a gram positive and a gram negative bacteria. After incubation of 12 hours, it was observed that the gram negative bacteria survived, whereas gram positive bacteria were killed. Which of the following is the possible mechanism of action of the given antibiotic? I need both the answers as well as the explanation. So just give it a random thought that what could have happened that only gram positive bacteria were killed and not the negative ones. D. How? Why? How? Both have peptidoglycan layer and it's kind of same. Yes, yes, I understand it's D. D. Okay. So peptidoglycan layer because penicillin has nothing to do with media or extra chromosomal DNA or like flagella of the bacteria. But I need the explanation like why? Why does this antibiotic act on peptidoglycan and why it differently acts on peptidoglycan layer in case of gram positive and gram negative? So suppose it's a particular antibiotic which affects the peptidoglycan layer. So why? Gram positive bacteria are more susceptible to antibiotic because of peptidoglycan layer. So gram negative bacteria also have peptidoglycan layer. So why were not they susceptible? Why are they more susceptible? Why? What makes gram positive bacteria to this extent susceptible? Yes, we have one more answer. No idea. Okay. Because gram negative bacteria has outer lipid layer which can protect them. Very good. Yes. The outer lipid layer is actually extra added protection to the gram negative bacteria. There's one more point which is important. You have seen that the periplasmic space between the peptidoglycan layer and the inner membrane is quite wide, right? That space has a lot of and beta lactamase enzymes which are secreted by the bacteria that are capable of killing the uh, which are making the bacteria resistant to these antibiotics, so they are not killed. So there are many mechanisms that because of this periplasmic space which are very thick, they have different secretions out of it, and these secreted antibiotics, the secreted proteins can destroy the beta lactam ring of many peptides, many antibiotics. And you know that most of the antibiotics, like penicillin, if I give an example, penicillin has this beta lactam ring which acts on the transpeptidase enzyme of the peptidoglycan layer, right? So, to inhibit that, bacteria has evolved in a way so that it can affect the beta lactam ring of the penicillin and, in a way, like not allow it to grow. So, yes, that is it. Does anyone else have any other opinion that why more bacteria, why is more susceptible gram positive and not gram negative? And one more thing, as you mentioned that it protects it, so how does this outer lipid layer exactly protect it? So what is the composition of this outer lipid layer, or what are the constituents of the outer lipid layer that it gives it advantage, like added advantage to the bacteria? Any idea? Anyone? So what happens is, because gram negative bacteria has which no, okay. So when we, I don't know if you have idea about the resistant mechanisms, how a bacteria becomes resistant to a, this outer layer has different porins or channels or water pumps or efflux pumps. What happens is the bacteria, the antibiotic gets inside the bacteria and it's immediately pumped out of it. It doesn't have any inactive, it doesn't need to be inactivated also. So this outer layer, some way or the other, has evolved through mutations to have specific features that inactivate the antibiotic, or maybe it selectively doesn't even allow it to enter. That is how it poses a specific help to it, like how it helps it to do it. Okay, so is that clear to all of you? In a minute, this session will get over, so please kindly leave and rejoin the session once again for the last segment. So I guess this was clear that why it's peptidoglycan layer. Let's move on to the next question. So here is a schematic of a type of DNA replication hypothesis. Which of the following statement is correct for the shown image? So what kind of replication process or hypothesis is this? So if I just brief it, these are the two parental strands, and in the after replication, this is again the parental strand, which is the now the daughter one, like the daughter one resembles the parent, and this is a complete different one. And again in the next one, only one is there, and these three are the different ones. So what mode of replication is this? Okay. Yes, it is a conservative. So let's just see that what are the three different models that were proposed and ultimately which one was used. So one of the models was semi-conservative model. So you see, there's a parent, two of them get separated, and then again using it as a template, a second strand is generated. So after that, again they are separated and another strand is generated. So we see that here it's a mixture of the parental and the new strand, and here again you have a segregated parental mixture, segregated parental, and so this is happening in a semi-conservative manner. So what is a conservative? Now let's come to what is a conservative. So in case of conservative, you see there's no mixture. Like from parent, a different new one is being formed. The parental strand is completely conserved here as well as here, like all the places. So if we compare it with the parental and the normal strand, it is happening in a conserved way. Like whatever was in the beginning, in the end also the same thing is there. But if it is a semi-conserved, then half of it is always there. Like you see, half mixture, half mixture. Again, in the next generation, what will happen? Then one will be blue and one will be red. Let's try to do that. So this blue, there will be the blue, and then again there will be the, I don't have pink, so I'll just put it like this. And this red will get separated. This blue will get separated. There will be a red and there will be a blue on it. So you see, it will happen in a semi-conserved way, right? So this is a semi-conserved way. And what is a dispersion model? That parts of it will be dispersed in the next generation. Like it's not completely blue or completely pink, like the semi-conserved way. Only these parts you see have been dispersed in the next cycles. Okay. So that is why it's called a semi-conserved way. So can somebody tell me which model is actually the accepted like model right now? Which one is the accepted model of DNA replication? A. That is a semi-conserved. You're right. So that was about the semi-conserved and the uh, conserved. Okay. So this question is also done. Now coming to the final question. Now this will need a bit of solving. So we'll all solve together. So what does this question say? The question says that the mice black coat color B is dominant over brown. So the dominant one is B. So, in order to be black in terms of phenotype, what will be the genotypes? VB or B small b, right? Now, so again, solid is dominant. Okay. So, in order to be solid, it should be S capital S or S even small s. So that is how the dominant and the recessiveness, I guess it's okay. So color and spotting are controlled by genes that independently assort. So there will be independent assort. A homozygous black spotted mouse is crossed with a homozygous brown solid mouse. So how will a homozygous black spotted mouse look? Homozygous black spotted means BB and a spotted means white spotted. So SS, because small s is recessive, so it has to be SS. It cannot be capital S and there. And again, it's in a homozygous brown. So small and solid. Now solid will be capital S because it's mentioned it's homozygous. Okay. So we are crossing this. Now, and also here also it's homozygous. So there's no other question of anything else. Both of them are homozygous. And both the traits are homozygous in its own condition. This is the parental generation. And in the F1 generation, they are saying they're black and solid. Now, by black, what we mean? It could be BB or BB. And by solid, what we mean? That it could be SS or SS. We are not sure about it. It's black and solid. Okay. Now we need to find that all generations are black and solid. The solid dominant over. Now we need to find that a test cross is carried out by mating this one with a brown and a spotted one. Brown means brown and spotted. Now we need to see that what could be the possible genotype of the parents and as well as the F1 generation. Now the genotype of the parents it's already given in the question. This one is the genotype of the question. So this and this, if I'm not wrong. And then let's see how F1 will be found out using a back cross. Test cross, sorry. Yes. So it is a black and solid. It could be this one, capital B, small capital S, capital S. So now let's see the scenario that if it was completely homozygous dominant, then what would have been the genotype? So the gametes would be BS and small b, small s. It would give a mixture, like it would give a heterozygous condition. So for it to be a dominant homozygous, the test cross should give me either dominant hetero or homozygous dominant phenotype. That is this one. Okay. That is what it is doing actually. That on crossing this and this, we are getting this. So it was a dominant homo. That is this one. The F1 generation. It was B, B, S, S. Clear? Now, if this was of the second option, that it was not a true dominant homozygous, it was a heterozygous dominant. So what will happen if it was a heterozygous? Then it's capital B small b, capital S small s, and cross with this. This is quite common, and this is how these are the 4 different genotypes we'll be getting in all 1, 1, 1 in 1, 1 ratio. Okay. Why? Because you see there's a variation here. That will give you. So, if my result gives me a mixed phenotype, then it is a heterozygous. And if my result gives me a dominant phenotype, either complete hetero, either complete homo or complete hetero, then like a dominant phenotype in one, it could be either completely hetero or homo, not a mixture like this. Then This will be a dominant homo. Okay, that will be the genotype. So let's get back to the question and see what was it, what my options. My options are these. You see, all the options are same. This is how I'm getting that what was my F1 generation genotype. It was a hetero one. We only got hetero ones. So it's a hetero one. A true. Let me get back and discuss. Let me discuss this one again. So if they were completely homozygous dominant, then they will give me heterozygous genotype. But if it is a heterozygous dominant, it will give me a mixture of both hetero and homo. Okay. So what I'm getting this one. And this is the genotype, the different ratio in which they are present. So this will be the answer. Parental generations were easy. Why? Because we got them both. It was already mentioned in the question that it's a homozygous. So the parental generation determining that was easy. But in case of F1, we had to see both the chances that either because it's a black, it could be a complete homozygous or a heterozygous. And we saw that because a homozygous one is giving me a pure, like it's giving me only hetero, so it's a homozygous one. Now, the second question is, yes, analyze the test cross results and select the correct genotypes and their expected ratios. So on analyzing, what did we find? That this was the test cross between, now we know that, okay, this was my genotype, and then I analyzed the test cross with this one completely along with the recessive one, and I found that all these were present in half hour. So if I just so you see BBSS, capital B small b SS, this one, capital B small is SS, this and this, BBSS and this one in the ratio of 1:1. Is it clear to all of you that how we got it? Hmm. Okay. Uh, there's one more question that came up in the discussion forum that from Arushi, I guess, about mapping. Like how I would try to just briefly say that how we do these mapping things. So the concept is higher the recombination frequency, the more far they are. So whichever has the highest recombination frequency, this is addressed to Arushi if you're listening. So the higher, is my screen visible? Just one second. Uh-huh. Yes. So higher the recombination frequency, far away are the genes. So like D and B were 33 units far away. So you just place B and D at the farthest, like this, okay? And then try to place in the ones which were there. Now you have to see that you have to place A here or A here. So is she in live? Then I would have asked her to copy paste the question. If she's not in the meeting. So I'll just open the question. Just give me 1 second. All of you. Yes. So the question said it's on linkage mapping and like determining the sequence of chromosome. So it was said that A and B are in a distance of 8 map units. Similarly, A and C have a distance of 28, A and D 25, B and C 20, B and D 33. So in order to solve this, what we have to do first is see that which is the farthest. So B and D. So you place B and D at the extreme edge. Okay, 33. Now you try to incorporate A, B, and C within it. So first thing that comes to my mind is the next farthest one is A and D. So I will place A here first. So that will give me 25 units. And then B and C are like 20 units. Now let me solve the A thing completely. A and B are of 8 units. So this clears my thing that, but now what comes is A and C is 28, whereas B and C is 20. So how should I place them? A and C farthest, and in between B should be there. So can C be here? Hmm. Can C be here? Is it possible? So what did I do first? First I placed A and B and D because they are the farthest one. Then I placed A in between it because A and D also have somehow 25 map units. And then I wanted to solve it for A and C and B and C. So A and C 28, A and C you placed it far away, and B and C 20. So you placed D between it. Now you just incorporate this here. What will happen is D, A, B and C. That is how you solve it. Okay. That is how you solve. I hope your doubt is clear. I've addressed this, so your doubt is clear here. So, does anyone have any other question regarding the assignments or like something that you want to discuss or some concept? If I'm able to say help you out, anyone? Or you find any particular question a bit confusing in the assignment? Is there anything else you want to do? Want to discuss? Okay, I just check if we have missed any questions or not today. Okay. So I guess we're done with today's session, and keep posting your questions in the discussion forum, and we'll try to I just stop sharing this. Yes. And I'll try to address them as much as possible in the live sessions. And okay. So thank you for joining today's session and see you in the next week.